Good afternoon, everyone. Thank you for joining us. It's my great pleasure to welcome you to this Marine Environmental DNA Workshop, which is co-organized by the French Research Institute for the Exploitation of the Sea, Ifremer, the Tara-Ocean Foundation, and the European Molecular Biology Laboratory, EMBL. My name is Bruno Hernand. I am an evolutionary biologist working at Ifremer and a member of the organizing committee of this event. On behalf of the organizing committee, we want to thank you warmly for your interest in this workshop and your responsiveness despite the pandemic. Unfortunately, because of the COVID-19 crisis, we had to turn this event into a fully virtual one, but on the other hand, this allowed us to increase the number of participants, and we are very pleased to have such a large attendance. We will actually be slightly above 100 participants throughout these 3 days. Environmental DNA and its use for addressing biodiversity research and monitoring is a rapidly expanding field that has already proven very successful, especially in terrestrial and freshwater ecosystems. However, our feeling is that the application of environmental DNA in the marine environment still poses several specific challenges from the technological, methodological, and analytical point of view, and also that its potential in terms of research questions addressed is far from being fully exploited. So his feeling is reflected in the thematic sessions of this workshop that will first try to give an overview of the field in several types of ecosystems. Then we will focus on sampling, methodological and analytical issues before opening up to promising avenues of application. Each of these thematic sessions will also be followed by short, half-an-hour-long brainstorming sessions organized in subgroups to foster discussion and the emergence of new ideas. So we are very honored and glad that 24 internationally renowned speakers accepted our invitation to cover these different topics and to get involved in the brainstorming sessions. I also want to address a special thanks to our speakers from the USA who accepted to participate despite the fact that it is very early morning for them at the moment. But without waiting more, I will now pass the floor to Dr. François Houllier, who is Chief Executive Officer of Ifremer, and then to Romain Troublé, Executive Director of Tara-Ocean, and Dr. Chris Boller, who is Scientific Coordinator of Tara-Ocean, and finally to Dr. Edith Erb, Director General of EMBL, for some introductory words from the 3 organizing institutes. So please, François Houllier, the floor is yours.
Okay, so it works. So thank you, Bruno, and good afternoon to everybody. I'm positively impressed by the large attendance of this workshop. Of course, we would have preferred, as you said, that it would have been organized in presence rather than using this system. I would also like to extend my sincere thanks and congratulations to the organizers. Indeed, it happens that we have had several discussions in the last few years on the Ifremer side, both with Tara-Ocean. We had a seminar, we had discussions, and we wanted to develop our collaboration. We also had talks with the EMBL last year, and we also wanted to do things together. So I'm very happy that the organizers have been able to draft this program and to assemble so many people, not only from Ifremer, EMBL, and Tara-Ocean, but also more largely from the scientific community to discuss the scientific issues related to marine environmental DNA. From my point of view, the goal of this workshop is obviously for you to share the state of the knowledge, the state of the concepts, and also understood what you said, Bruno, about the fact that there are some technological difficulties or things to be solved. So I understand that there are a broad range of issues that you want to discuss, not only science, but also the technique, the practices, so that there are future progress in this area of marine environmental DNA. So it's good that you do that. It's also good that you identify new research questions and the different technological points that have to be solved. On our side at Ifremer, we are also very much interested in that this workshop results in the end in new projects that can be developed at Ifremer, of course, but also in close relationship with the community of Tara Ocean, with EMBL. Probably Edith will say it. We plan to have a MOU between the EMBL and Ifremer, so I think that's this workshop is most welcome for that. In the end, I would like to say a few things. I'm not a specialist of marine environmental DNA, but there are at least 3 areas in which it's important for Ifremer to progress in this domain. We have a mission to survey the coastal marine environment, and we have been having for a long period now a lot of networks to do this survey, and I do believe that marine environmental DNA will be one of the future technologies to continue this survey and this mission that we do have, not only for science but also for the sake of the different ministerial departments. The second area for which it is important is the evaluation of fish resources, fish populations. I never use the word fish stocks, which I do not like at all. So fish resources, fish populations. And here again, I understand that we have started doing some things at Ifremer, but I feel that I would like very much that at the end of this workshop, you tell me more. I mean, Bruno and the other colleagues about the potential of these techniques, not immediately, but in the future. And the last area, which is key for Ifremer, is probably definitely the understanding, the characterization of the deep sea environments. So I think in these 3 domains, the surveillance of marine coastal ecosystems, the evaluation of fish resources, and the knowledge of diversity of deep sea environments, there is a lot of things that can be done with a marine environmental DNA. With that, I don't want to be much longer. I wish you a very fruitful workshop, and I do expect that at the end, some way or the other, Bruno and the other organizers, we have an opportunity to meet, and you tell me, so what are nice things that we want to do with these techniques and these methods. Thank you.
Thank you very much. I will now pass the floor to Romain Troublet.
Hello, everyone. I'm very pleased to be here today. Thank you, Francois, for your introduction. And I would also like to thank the organizers, the team who organized this event. And this indeed has started a 1.5 years ago when we met with Ifremer and Tara Ocean folks in Paris. It's a nice development on what we have planned to do, and it's coming up now despite the COVID situation, which is really great, even if it's remotely done. When I look back into the past of what we did with Tara Oceans and the Tara Ocean consortiums and EMBL and CNRS, we launched this program in 2008. At this time, DNA technologies were far out of reach for ecology, and all these scientific approaches by far too expensive. And in a hand of years, I mean, in 5 years later, what was costing $1 billion to sequence and to use this technology to tackle the ecological aspect of the ocean, it became 10 millions, and then since then it's also again evolving so fast. And I'm impressed also every day we launch programs with the Tara-Ocean folks and Tara-Ocean Foundation. The technology is moving very fast, and the size of reach is moving very fast. The cost, the speed of it, and when we back in the 10 years ago, we did study with some samples of the plankton ecosystem in the ocean. On the last expedition in the Pacific and the coral reef, I think we managed to sequence and to use the DNA of 10 times or 20 times more than previously done on the plankton. So very happy to be here to listen to the ideas and to also the bottlenecks we can see here in this field, and I'm looking forward for the next step. Thank you very much.
Thanks a lot. Chris, if you want to say some words, Chris Bowler.
Yes, I will do. Thank you, Romain. So also on behalf of the Tara-Oceans team, I'd like to say a collective thank you for organizing this event. It means a lot to us. It means a lot to the foundation. As Romain has mentioned, it means a lot to the consortium and the scientists working around Tara in general. I am now scientific director of the Tara-Oceans Consortium, taking over from Eric Calcenti, who's been at the helm since 2008, really working with Tara, setting up the Tara-Oceans program, and that was very much the baseline to sort of establish omics methods, DNA-based methods, in the research being performed by Tara. As Romain mentioned, when we started in 2009, we had no clue of where we would go because the costs were prohibitive for doing, what we would like to have done in our dreams. The sensitivity of DNA sequencing was not there yet. And already over this last 10 years, we have seen the incredible progress that has been made in the field of DNA sequencing technology and omics in general. Romain didn't mention, but now there's been several times when we've actually been sequencing DNA on board Tara using the ONT technology, and obviously it works, right? So being able to move from a lab setting to a ship setting for sequencing your DNA really opens up a lot of perspectives in terms of doing reactive work on board a ship to modify your sampling program to deal with what is actually happening in the water around you. This is really, quite a technological paradigm shift in the field. And I guess this change in technology, progress in technology is going to continue forward as we think more about, considering sequencing DNA actually in the water column through automated techniques and so on. So this is really definitely going to be something that is going to continue for the next several decades. And as has been mentioned already, it's very exciting technology. It's a really important technology. There's lots of potential opportunities, but there's also challenges, there's caveats, there's a lot of issues that we need to consider to ensure really the best operating standards, to try and get as close as we can to quantifying the DNA that we detect in the environment. And I think this is the collective work of the community that is required to move those standards forward to ensure best operating principles as soon as possible. So we're very happy that everybody is here. I think we're going to have a very exciting 3 days of open discussion together to try and consider these very important topics. So I'll end there and pass the microphone back to Bruno. Thank you.
Thank you very much. I don't know whether Edith Erd could join us. I don't think so.
She's not there yet, but I'm sure she will come in a minute.
Yeah. So maybe I can start saying a few words about organization, and I can pass her the floor when she joins. So in terms of organizational matters, first, before starting the thematic session, I will remind you a few things. First, so let me remind you that the talks will be streamed live on YouTube and available as replay for 1 month, except for those speakers who explicitly said they didn't want to be streamed online, so there is no problem in that respect. Let me also let you know that the links will anyway not be referenced, so that the stream will not be available to people having received them. Sorry, I'm losing the track here. So I was saying the links will not be referenced, so they will only be available to people who received them directly from us. So as you might have already noticed during the talks, your microphones will be automatically muted. You will be able to open them again for questions after each talks. And you will also be able to open them for the 10 minutes, which will be dedicated to final questions at the end of each thematic session. So for asking questions to the speakers, we ask you to raise your hand, either by using the corresponding icon that you can find at the bottom right of your screen in the EMO icon menu, or at the bottom of the participant list when you open it. It depends on the version of the Zoom software that you are using. Then the chair of the session will give you the flow based on the order of appearance of the raised hands. And also let me warn you that as for the talks, the questions will be streamed also online. So if you do not want to appear on the videos, you can simply write your question in the chat and the chair will read it. As I already mentioned, we will have also, after each thematic session, half an hour long, roughly, brainstorming session. For this, the attendance will be automatically split into a number of subgroups, which is equal to the number of keynote speakers of the corresponding thematic session. And the discussion in the subgroups will be animated by a member of the organizing committee with the help of a rapporteur, and it will draw on the keynote speaker's expertise. So as for the question sessions, your microphones will be operational again during the brainstorming session, and we ask you to proceed as for the questions after the talks, that is by raising your hand and waiting for the member of the organizing committee to give you the floor. In that case, for the brainstorming sessions, there will be no live stream, so there's no problem of video recording and so on. On the same occasion, so during the brainstorming sessions, you will have whiteboards available and accessible to all the participants of the subgroup, so that in addition to oral participation to the brainstorming, you can also contribute to the discussion through written notes. I will, I think, show how these whiteboards are accessed and how they work just before the first brainstorming session. We will take 1 or 2 minutes to show you that. Finally, I can mention that there is a specific chat that is dedicated to the workshop that you can find on the dashboard. So maybe I can share my screen to show you this. So I hope you all see my screen. And so here on the dashboard web page, after scrolling down a bit, you will find click here to chat. So you just need to click on there and you will end up in a Discord-like chat, which is organized with channels according to the different sessions and where you can discuss topics related to the different sessions or ask questions. There will be also a general channel and a channel for announcement related to changes in the program. So this chat will be running for the 3 full days. So basically you can use it on the side of the different sessions. And of course you can use the Zoom chat during the sessions themselves. But you can use this Discord chat even in the mornings or in the evenings at your will. Okay. I stop sharing. Okay, I think that's all. I don't know whether Edith could join us for some introductory words.
I don't see her yet. I mean, in the meantime, I can of course welcome everybody in the name of EMBL, the European Molecular Biology Laboratory. I think you will be aware that we are also an intergovernmental organization with 27 member states across Europe, and certainly well known for basic research in molecular biology. And of course, we also have a mission in directing societal, working on societal issues. And traditionally, this has been health for EMBL, sort of the applied direction. And only very recently, and coming with our new program, we have opened up a new direction at EMBL that is also addressing, if you want, planetary health. We have dubbed this direction planetary biology, and with this direction would like to reach out and work on societal issues that concern the environment, also conduct research in the environment. And one big project that we have started in the context of this planetary biology program is the track expedition, Tara-EMBL Coastal Exploration. With this expedition, which is where we team up with Tara, EMBRC, and also Ifremer, we would like to sample diversity and investigate communities at all scales at the land-water interfaces along all European coasts. And this will happen in 23-24. And in the context of this expedition, it has been a pleasure for us that Ifremer and Tara opened up the organization of this workshop also for EMBL, so that we can reach out to new collaborators, as it will be very important, of course, for the success of this program, that we can collaborate and learn with as many experts in environmental sciences as possible. Part of this project will also be to explore eDNA, environmental DNA, for all projects relating to the sampling of communities and ecosystems. And this is why we are very happy to have joined the organizational team of this eDNA workshop to support it and to learn jointly with our collaborators on this very exciting topic. And I think I will leave it with these introductory words, and we continue then with Bruno.
Okay, thank you very much, Detlef. Pity that Edith could not join us. I will now hand over to Chris Bowler, who will chair the first thematic session, because it's time to move. So Chris, the floor is yours.
Okay, thank you, Bruno. And since Edith was unable to join us, let me just mention one thing that I forgot to mention in my introductory remarks, was that the DNA sequencing that we did within Tara Oceans and within Tara work in general has all been performed in Genoscope, who are the people who have been very much pushing the technology of DNA sequencing along. So I should have thanked Genoscope for their fantastic dedication to the Tara work, and I apologize for omitting that. So with that, then let's move on to the first session of the workshop, which I'm very pleased to chair. So this first section is on best practices, really taken from some state-of-the-art presentation, state-of-the-art work that is going on in the field. And we have three keynote presentations to kick off this session, really moving from ancient to modern DNA through metagenomics and metabarcoding based workflows. So I will move on and introduce the first speaker to you, who is Yucheng Wang. I'm very happy to introduce him. He's from the Department of Zoology at Cambridge in the UK, and he will be talking to us about ancient environmental metagenomics work. So Yucheng, I hope you're able to share your screen and keep going. I'll give each of the keynote speakers 15 minutes so that we have about 5 minutes of questions after each presentation. So I'll give you a bit of a nudge after 15, 16 or so minutes. Okay, Yusheng.
Yes.
Over to you.
Okay. Can you hear me well?
Yes.
Okay, great. All right. First, thank you very much for you guys for organizing this workshop, and thanks a lot for having me here today. My name is Yusheng Wang, and I'm currently doing a postdoc at the zoology department at the Cambridge University, and also holding a visiting postdoc position at Geogenetics Center at Copenhagen. My topic today will be ancient environmental metagenomics, explore the full potential. So we'll very briefly summarize the recent advances and application cases of using shotgun metagenomics on ancient environmental DNA, ancient eDNA, in our lab. All right, so since the first start of the ancient eDNA, there has been a steadily increase of the relative studies. But as you can tell from this figure here, the relative number of publications using ancient eDNA is smaller compared to using the eDNA for modern sightings. However, this small fraction of publications did set up some notable landmark studies of the eDNA field. This is mainly because when doing ancient eDNA, we are facing some extra problems. For example, we have extremely low DNA concentration in these ancient samples, and the DNA molecules are normally highly fragmented and damaged. Therefore, in order to deal with these extra problems, we have to use some more kind of complicated strategies, both in terms of the laboratory protocols as well as the bioinformatics pipelines. And this kind of a methodology and technical development in some ways also benefits the whole eDNA community, and maybe also potentially interest for the marine samples. So far, the main methods the main approach used for sequencing eDNA is the mite barcoding. Despite its high, efficiency, in species identification as well as high successful rates, it's only targeting a small region across the very large whole genome. For example, the most widely used plant primer for mite barcoding in ancient eDNA is this tRNLJH primer, which only targeting and amplifying a short marker gene around 100 base pair long across the genomes of different plants that can be, each of them can be billions base pair long. So in other words, mite barcoding in a way only exploits a small fraction, a tiny small fraction of the available DNA preserved in the environmental samples. An alternative approach is to using the shotgun metagenomics, which basically sequence all DNA across all organisms without any sort of a species-specific amplification or targeting. By doing shotgun metagenomics, we can see some very obvious advantage. First, because we sequence all organisms, the DNA of all organisms in one run, therefore we can have a higher efficiency for reconstruct the whole ecosystem across the whole tree of life. And because we sequence the DNA distributed across the whole genome of different species, therefore higher sensitivity and better taxonomy resolution can also be expected. And because we are not relying on PCR amplifying the short markers anymore, therefore an improved quantification of the identified taxa can also be expected. The last advantage I can think of, particularly for the ancient eDNA, is that we can using the DNA damage patterns to further authenticating the identified signal. And of course, it also comes with some disadvantages. It's not perfect, right? I'm not saying it's so. First, it will request whole genome level reference database because we are sequencing the whole genome level data. This kind of a reference database, however, is not available for majority of the regions across the world, and it will be more expensive. And because we have a much larger reference database as well as the dataset itself, therefore the data analysis will be more complicated and time-consuming. So there have been some successful applications of shotgun approach on the ancient eDNA samples. these two studies I'm listing here are considered as the very initial attempts, of which the first, the communities of higher organisms of plant and animal were directly reconstructed by shotgun sequencing the DNA preserved in the lake sediment across a series of lakes in Canada. And in the second case, the population dynamics of woolly mammoths was successfully reconstructed through shotgun sequencing the environment DNA preserved in lake sediment, again in lake sediments on the St. Paul Island. However, the application of shotgun approach are not always generating a very positive result. For example, this paper published on 2019, they found there's a very small fraction of overlaps between the taxa identified by DNA shotgun sequencing to the taxa identified by pollen and microfossil. This is most likely due to insufficient reference database. So our recent study found that the reference database is extremely important for the shotgun sequencing-based taxa identification. So in this study, we shotgun sequenced more than 500 samples across the whole Arctic and mapped all these reads against the 3 different reference databases, of which the DB2 is generally the public NCBI database. And for DB3, we further add the chloroplast, the genome of Arctic plants, into the DB2. And for DB4, we further again add the whole genome context of these Arctic plants into the DB3. So by mapping against all the Arctic reads against these 3 databases, we found that we have a dramatically increase of the identified reads into plants by at least more than 20 times. And the database coverage for the Arctic species also increased dramatically by many order of magnitudes. And another improvement of offering proper reference database for the shotgun data is to further decrease the proportion of the false positive identifications. As you can see from this figure on the right, where we have more and more reads identified into Arctic plant species instead of the false positive non-Arctic species. And the same case also occurred for the animal identification. Here's an example for the woolly mammoth identification. So based on the DB1, which is the NCBI database, we hardly identified any DNA into woolly mammoths from across these 500 Arctic sediment samples. Well, by adding the mammalian genomes into the reference database, we can well identify the woolly mammoths, this green color here. And another thing is that the previous reads false positively identified as African elephant, this green color here, were later also mapped onto mammoth DNA. Therefore, they merged into the higher taxonomy levels. In this case, it's the elephant family, which covers both species. So in short, the reference database will be the first thing to be considered if you really want to go to a shotgun approach for sequencing your eDNA. And we also found that higher diversity can be captured by shotgun approach. So in this case, we compared the identified plants from 131 overlap samples between this new Arctic shotgun datasite and the MIT barcoding datasite in 2014. Of course, the methodology, the whole methodology and technology has been improved a lot since 2014. But we found that between these 2 datasites, only except 2 samples, for all other samples, shotgun captured higher plant diversity than MIT barcodes. And quantification is a possibility now. So because in the lab for doing shotgun approach, we simply extract and sequence all DNA molecules literally. Therefore, there's a very rare chance to have bias introduced in the data generating. The main issue will therefore be the different efficiency of taxa identification pipeline for different taxa. Therefore, we simulated 1,000 data sites with known risk composition to investigate this difference of the identification efficiency. As you can tell from these 4 samples here, the identified risk is directly determined by the input risk for given species, for given plant species in this case. while the efficiency of identification can be very different for different taxa. And this efficiency is determined by the genomic distinctiveness of a given species rather than the reference data coverage for that species. That is, when we have a species whose genome is more distinctive than the genomes to the genomes of other species, we will have higher chance to correctly identify its DNA. So it offers a way to really quantifying the composition of the ecosystem. And another very important benefits of doing shotgun sequencing for ancient eDNA is that we can draw the DNA damage patterns. For those who are not familiar with ancient DNA, the damage pattern is some certain substitutions of the bases, which has a higher frequency to be happened in the ends of the DNA strands. So by showing, by calculating, by quantifying this substitution rates along the DNA strands, we can draw this damage pattern models, which offers an extra way for us to authenticating the identified ancient signal from the potential modern contaminations. As you can tell from these three figures on the right, E, F, G, where we can now draw perfect damage patterns through the sediment preserved ancient DNA, which is even better than the signal. The damage patterns can be resulted from the fossil samples. And we can also zoom into population levels or beyond the presence and absence data. For example, here is again the woolly mammoth example where we built up this phylogenetic tree of mitochondria, of mammoth mitochondria, both from environmental DNA as well as the fossil recovered genomes. As you can tell that the eDNA agrees very well with the mammoth mitochondrial phylogeny reconstructed from the mitochondrial genomes of fossils. Generally, the clade 1C represents a North American clade, while the clade 1DE represents a Siberia clade. And we also did the same for the horse for this intercontinental Arctic shotgun data site. And one particular interesting clade is this Yukon clade, which is very diverged from the whole phylogenetic tree of horse. And all the all the mitochondrial genomes for this clade were recovered from fossil samples distributed in a very small region of Yukon, Canada. And surprisingly, we found that all the 15 eDNA identified horse we successfully placed into clade are all from Yukon as well. And 10 of these 15 samples are from a site which is only 10 kilometers away from the location where 2 fossils of this clade were found. So this confirms the reliability of using ancient eDNA to really looking into the phylogeny rather than presence absence. Some more recent methodology advances. First is to apply the robotization in the lab. So as I mentioned, for generating the data, I have been talking about this Arctic data site. We spent almost a whole year in the lab for processing the sample for data generating. However, by using this new adapted robot system, processing this 500 sediment samples in the lab can only take 2-3 weeks now. But however, the robot system does not only mean a high throughput of sample processing capacity, it also means less potential contamination from the people who handling these samples in the lab, and also better standardizations. Therefore, we have a better chance to really compare the signals recovered from different samples. And another methodology advances is to apply the target genome capturing, which is basically a technique where you can capture the DNA of a particular species you are interested in out from the whole bunch of DNA from all other species. So in this paper published last week, the first ancient whole genome of a species was ever first reconstructed from directly from sediment samples. And all this kind of population genetics and administer figures, all this kind of evolutionary analysis can be done in the future on the sediment DNA on a whole genome level. So what I can think of for the near future directions for applying the shotgun approaches for particularly ancient eDNA. First, of course, is to the population genetics for individual species directly from the ancient eDNA. That is to draw the evolutionary history of this species we are interested in. This will be particularly important for species where we have rare or we have known fossils available for ancient DNA extraction. That is majority of the plants. And all this kind of microbes, all this kind of stuffs without good fossils preserved. And the second will be to profile the functional genes, functional traits. Therefore, we have a chance to really look into the complete ecosystem that covers all components from virus, bacteria to insects to plants to macrophones, as well as the the material and energy cycling based on these functional genes and the functional traits. And the third direction I can think of is to exploring the real unknown. So far, there's only up to 10% of the shotgun data can be well identified based on current methodology and the current reference database. That means we have at least 90% of the data remain really unknown for us to explore. That's my talk. Thank you very much for your listening.
Wonderful. Thank you very much. Let's see if my reaction works. Yes. Great. Thank you, Yu-Cheng, very much for this really great overview of where we're at with ancient DNA. Again, the advances in technology is amazing. What now becomes possible to do. Do we have any questions from the audience before I? Pierre Borg, yes, please.
Yes, hi, great talk. I just want to question, you have this degeneration of the DNA and then conclude that was ancient DNA. Is there any chance to get the timing out as well? So amount of a certain kind of modifications to DNA gives you an idea when the beast has been alive, or do you have rely on external data?
Basically the current method is really relying on the radiocarbon dating and OSL dating for specific sediment layers, right? So we have to rely on other methodologies to determine the ages of our samples. Well, if you are talking about the damage, that is something that we are really trying to use that signals to use those models to determine the age of this DNA. But I mean, I would say it's still far away from a conclusion to really relying on the DNA itself.
Thanks.
Okay. And Detlev, I believe you have a question too.
Sorry, I was still muted. Yes, thank you very much, Yu-Cheng, for this wonderful presentation. I was wondering about the time horizon. So if you have these layers, where does it stop? So is there something like in a abrupt end rather, or does it slowly fade out? And at what time horizons are your limit? Yeah, so well, the current limit for ancient DNA is about 1.6 million years, right? But it's from the horse covered in Siberia in the permafrost. Well, for the ancient DNA, for ancient eDNA, I think the current limit is I don't know, it's probably some hundreds of thousands of hundreds years. So yeah, we have a limit for recovering the ancient DNA from sediments. And is that limit currently technical or due to history? So sorry, I didn't.
get.
Is it limit technical, or so can be overcome, or is it due to?
Yeah, well, so it's a complicated question, right? So well, it's technical limits, because when the sample really get old, the DNA molecules will be extremely short, right? So currently, what we are mainly working on, which can make sense, is DNA molecules, around some 40, 50 base pairs long. Well, when you really go to very deep layers, you can have, very, very short DNA, which makes no kind of an ecological or taxonomy sense, because it can map to everything. It's too short for us to really working on.
Okay.
Yeah, that's, I guess, yeah.
Thank you.
Okay, thanks, Yusheng. Maybe we should move on in the interest of time, and there will be a additional question time at the end of the session, so save up your questions for that if you didn't get a chance already. So let me move on to the second keynote. I'm happy to welcome Philip Francis Thompson to you, coming from Aarhus University in Denmark, who's
that's where we see the spikes in environmental DNA.
Final keynote then from Pierre Taberlet from Grenoble in France, who's going to talk to us about best practices in metabarcoding. And Pierre, I think you're in a very good position to tell us about best practices because you perhaps have more experience than anybody else in the field. So great to have you with us.
Okay, thank you for inviting me first. So, and thank you, Chris, for the nice introduction. So today I'm going to give a talk about how to avoid big mistakes, I would say, okay, in DNA metabarcoding. So I don't know if you know this kind of curve. This is the promise of an emerging technology in an industrial context. This is a Gartner Hype Cycle, okay? So when you have a new technology, then you don't know, so you start having expectation, and then you have a lot of expectation suddenly. Then you get depressed because you realize that it's not so easy to implement, and then slowly to implement, you improve and implement really the new technology. It's exactly the same in science, okay? But it's interesting to know that, for example, when you are here in science, there is a risk of publishing wrong results due to inflated expectations and publication pressure. Okay, so this is well known for ancient DNA. For example, we were talking about that 2 talks ago. So, you know, it was published dinosaur DNA in leading journals. It was publishing insect in amber DNA also in leading journals, and everything was wrong. Okay, so there is a real risk. And with environmental DNA, I think that we pass maybe this stage, but it's true that at some stage, people were just thinking that it was very easy and that you can do anything with environmental DNA. So this is not true. So my talk today will focus on the implementation of best practices to try to get good results. Okay, so I will focus on this improvement here. So, a few years, so we were several editors of molecular ecology 2 or 3 years ago, and we had to reject manuscript without review because the experimental design was really not appropriate. And then all the authors were complaining, telling us that with the same experimental procedure, this paper has been published in a leading journal. Okay, so we decided to write an editorial for molecular ecology, trying to list the best practices. Okay, and we associated a lot of colleagues that are doing a good job in environmental DNA. And I think that based on this editorial, we were able to reject some manuscripts without reviews if they were not okay. So there is a paradox in metabarcoding because when you look at the different steps, you have the sample collection that is quite easy. DNA extraction is easy. Amplification also, everybody is able to amplify DNA now. Sequencing, you just have to pay and you get your sequences. And the bioinformatics, there is a lot of pipeline to analyze the results. So basically, each step seems to be very easy, but I would say that it's very difficult to set up a sound experimental design. So there is a kind of paradox here because every individual step you think that it's easy, but actually this is not. And you can make mistake at every step of the process. So I will mainly focus on a sample collection. So, and first I would like to draw your attention on this paper. I like it very much. You know, it was published in the field of microbiology, working with DNA. And the title is Replicate or Lie. So if you don't have biological replicates, for example, you cannot compare samples because you don't know if you are just analyzing noise or if you are analyzing the true signal. So this is very, very important. And I think that this is true for biological replicates, but also for technical replicates. I will talk a little bit about that later. So when you, so the sampling is really the key step, and Philippe was talking about that very recently. So sampling is the key step. And if you make a mistake at the sampling stage, everything, all the rest of the process is useless, you know? So this is very important to secure the sampling strategy. So you are at risk of undersampling. For example, if you sample just randomly 5% of the community you are working on, so how to compare sample when you took randomly 5% of the species you are in your communities? So you are at risk also of contamination. You can also contaminate if you don't clean your sampling tools, for example, properly. And the controls you can do is quite simple. You can have pilot experiments. So I really also, sometimes we receive manuscripts in molecular ecology that are just pilot experiments that are not the real study, okay? And this is really true. So you should carry out a pilot experiment mainly for securing the sampling strategy. You must be sure that you capture the local biodiversity of your target taxonomic group by using your sampling procedure. And this is not obvious. For example, in soil, if you take 1 gram of soil, you have very, very few chance of getting earthworms. To get earthworms, you need to extract and to homogenize using at least 1 kilogram of soil, I would say. Okay? So this is one of the main difficulty. And I will illustrate also the complexity of the problem. So I don't speak about the timing as Philippe has done. I just will talk about one study that has been carried out by Raphael Sivad a few years ago. He wanted to sample fish in rivers. And at that time, he didn't know how many capsules for filtration. And you can filter 30 to 50 liters by each capsule. Okay, how many capsules he should use to capture the fish biodiversity in these different rivers. So he decided to use 11 capsules plus 1 for the control. Okay, 12 is nice because you know the 96 plates. It helps for the next step of the experiments. So, and what he found is that if you look at the number of capsules of samples you need to reach 95% of the fish diversity, you can see that according to the different rivers, you don't need the same number of samples. So this means that when you have to design a large-scale study, you cannot have 8 capsules for all the different sites, but you need to decide. And obviously, this is a compromise. Okay. So for example, in our studies we are carrying out now, we are using 3 capsules. Some others are using only 2. But we know that we might not be capturing all the diversity we wanted to capture. Okay. Also, I just give you an overview of some of the recent improvements we have done concerning sampling from water. So traditionally, if you wanted to use a filtration capsule, people were using a peristaltic pump. We are using a peristaltic pump and a disposable tube, because if you reuse the tube, you will cross-contaminate your capsules. Okay, so you have to change the tubing. So we opted for a different approach. We put the capsule directly into the water. We have the tube, and we use a membrane pump by sucking the water. And like that, actually, this is working very well. So you can have a look to the system we are using in the field. So it's a very homemade system, you know, at the moment, but we'll improve at that level. But you can see that we have a pole with the capsules, battery and pumps, and we have buckets to measure the volume we are able to filter. So we are filling the buckets 5 times. So this is because we decided to filter 50 liters per capsule. So here are the 3 capsules we are sampling at the same time. So it's quite convenient. Ah, yes. So, but I wanted also to mention something after Philippe's talk. I am not sure that this capsule will capture the same biodiversity than this one. This needs to be checked, you know. So there is a time differences, but there is also some spatial differences, and this is quite important. So this means that if you have a single sample per locality at once like that, so maybe it will be not enough. You need really to have several samples from the same location. So this is the pumps we are using. So these pumps are very cheap, 30 euros or less, even less, I think. So this is the whole system. So we are already using that quite a lot now, and this is we are quite happy. So if somebody is interested to get more information, just send me an email and I can provide you with all details. Also, we decided to have a look to the capsules we need to use. So I know that Philippe is a fan of the Sterivex. So no, but actually we decided to compare these different types of capsules, but with a constraint of using a very simple DNA extraction later on to simplify the work in the lab. Okay, so and what we have got is that the Sterivex was not working very well for fish with our simple extraction procedure. But I know that with a complex extraction procedure, you can get good results. But these 3 capsules were working exactly in the same way. Okay, so and as the cost was quite different, we decided to continue with the Watera capsules. And the Watera, if you contact this company in France, you can get the Watera capsules for 18 euros, which is close to the price of a Sterivex actually, but you can filter 50 liters. The protocol we are using, we carry out the filtration in the feed. Then we add 50 milliliters of buffer, Longmire buffer, that is well known for this type of thing, but without SDS, is to avoid to release DNA into the water. Then, very important step is vibration at 20,000 vibration per minute for 1 minute. We realize that this is a key step that need to be standardized. If you don't agitate the capsule in the same way, then you don't get back the target DNA that is stuck on the membrane. Okay, and then we transfer immediately the buffer into a Falcon tube. So this means that previously we were keeping the buffer into the capsule, but we try to avoid that because it's much simple after the vibration to get back the capsule. And then in the lab, it's very, very simple. It's only for cell remains, you know. We centrifuge the Falcon tube, we discard the supernatant, and we perform a very simple collagen extraction, for example, for tissue on the pellet. So it's very, very simple. So if you need information also about that, just don't hesitate to contact me. So DNA extraction now. So I will not spend too much time on that. Also, there is some risk, mainly risk of contamination. Okay, and you need to know that according to the extraction kits you are using, you can get different results. This is very well known for people working on soil because the lysis step is crucial in this area. I don't know if for marine stuff this is the same, but probably if the lysis step is also important, you might get, I think, different results according to the lysis procedure. Yes, and also at this stage, you need also to include some technical replicates, some negative and positive controls. So DNA amplification. So as you know, you can have polymerase PCR errors. Okay, so I will start, yes, with this polymerase errors. So personally, we recommend using a proofreading polymerase. but I know that there is no consensus. This is a controversial area. And the problem with proofreading enzyme is that the proofreading enzyme, if there is a mismatch on the 3' end of the primer, the proofreading will eat the 3' end of the primer and extend it again. So this means that you lose the 3' specificity of your primers, okay, with proofreading, except if you use the phosphorothioate primers, but as it's quite expensive, so usually nobody is using phosphorothioate primers. And then you generate a lot of extra bonds, extra sequences that you don't want to have in your results. So, okay, so proofreadings make less mistakes during the replication, but introduce a lot of non-target sequences. Yes, also. And also, there is the possibility also to use inappropriate primers. I am very surprised to see that in many metabarcoding papers, even the primer sequence is not given. Recently, I had to edit a paper where they said that we use the primers published by somebody, okay? It was a list of maybe 20 primers in this paper, but they don't mention which one they were using. But it's clearly the results depend on the primers. If you use different primers, you will not get the same results. Okay, and also there is a controversy between using short or 12S, 16S primers or CO1 primers. So it depends on your goal actually. So sequencing, there is a lot of problems with library preparation for sequencing. So obviously there is sequencing errors, but you can quantify it. You can know what is going on at that level. But the most important when you are using the Illumina sequencers is that you can have tag jumps and index mistakes, I would say. So tag jumps, this is a short sequence that you put on the 5' end of your primers, okay? And when you prepare the library, there is a few mixing among samples, okay? So, and this is quite problematic because this means that you misassign some sequence to the wrong sample. So if you don't try to monitor this type of tag jump problem, then you don't know how to filter properly at the bioinformatic level. So there is 2 papers dealing with the tag jumps that has been published by Christine Bowman mainly. Okay, and this one, TagSTD. So this is a protocol to limit, not to avoid, but to limit the impact of the tag jumps. And also, if I go back to this, so you know the index of the Illumina adapters that is used to identify the library. So sometimes there is double indexing, so it's much better in this case. But about 1 sequence out of 1,000 is misassigned to the wrong library. So this means that if you sequence in the same lane 2 different libraries with the same primers, you are at risk of having some results from the other library into the one you are working on. And you need to know that. And this is not negligible because as you are getting millions of reads, so if you have 1 million reads, you know that 1,000 reads will be from the other libraries, basically.
Could you start to wind up now? Thanks, Pierre, in a minute or so.
Yes, okay, so I can go very quickly with bioinformatics because there is a lot of pipeline for that. Okay, so I just summarize. So I suggest to carry out pilot experiments when it's necessary, to have enough biological replicates, enough technical replicates, many extraction controls to detect potential sporadic contamination. If the contamination are sporadic, then with one control, it will not be enough. You need 10 or 20 negative controls. Okay, this is the same with the negative controls. You need to estimate the tag jumps and maybe index jumps if relevant. So thank you for your attention and sorry for taking too much time.
Thanks, Pierre. That was great. Yeah. Yeah, we're running a little behind, but does anybody have a question quickly before we move to the breakout groups? If not, maybe I can just ask a quick question specifically for you, Pierre. What is your view of where the future lies? You know, people seem to be more and more enthusiastic about shotgun approaches, so avoiding the whole PCR amplification issue, primer issue, different artifacts from those depths. Do you think shotgun is going to be the future?
I think that this is a difficult question. So yes, for sure, for some of the problems, some of the scientific questions, the shotgun will be the definitive approach, especially for ancient DNA, because you can validate your ancient sequences by the modification at the end of each product. Okay. The capture might also be a solution for using the better way, the CO1 reference database, for example. But I am almost sure that for the routine monitoring, the classical metabarcoding, the PCR-based metabarcoding will stay for quite a long time. Maybe adapted, and I am not sure that we will stay with Illumina sequencing. We might switch to nanopore sequencing because you can get the results almost immediately. So, but as soon as they will improve the error rates, yes, we might switch to nanopore sequencing. So this is more or less what I see. So this means that there is no clear answer. On one side, for research in ancient DNA, for sure, the shotgun sequencing will be helpful. The capture might be helpful, but there is also some tricks, I think. And I think that the PCR-based metabarcoding will stay for a long time for the monitoring aspects.
Excellent. Okay. So I think I know that the web platform we're using is very strict on time. So I think the coffee break is going to be precisely at 4 p.m. So I think maybe we will end the session here, and I think we should all now migrate automatically to the 3 breakout rooms. So if I understand correctly, Bruno, correct me if I'm wrong, just stay online and we will move, you will be migrated automatically to one of the 3 rooms to continue the discussion.
Yeah, maybe I can step in. There's a raised hand from Colomban. Maybe we can give him the floor just for 1 minute, and then I can just show how the whiteboards are working before we split.
Okay.
We don't hear you, Colomban. Your mic is not on.
We are so lucky to have Pierre, so I just need to ask him a question. Pierre, what do you think? How could we improve the quantification of barcodes? What are the methods to make it a bit quantifiable?
So this is an excellent question. So we are working on that. We are getting relatively nice results, I would say, by simulations, and we are in the process of doing some experiments to validate what we get from. That's okay. So for sure, this is very important also to work on this aspect. And for sure, CO1 is not a good choice if you want to assess quantification. You should use a primer system that are much more conserved. Okay, but yes, this is on the new version of our book, we will add a chapter on the quantitative aspects.
Okay. Do you use a sort of etalon, you know, of
Yes, obviously there is, yes, obviously it will be based on spiking the PCR. So the ideal would be to spike the sample, but then you don't know what is going on with DNA degradation or stuff like that. So it's difficult to spike exactly in the same way different samples, but you can spike properly the PCR, I think.
Thank you.
There is already a few papers dealing with this aspect.
Okay, thank you very much. So just before we split, as I mentioned in the introduction, we will have access to the different brainstorming groups will have access to a whiteboard. So I can just show you how it works. I'm sharing my screen. I hope you see it. So this is how it looks like. So basically the people animating the brainstorming groups will provide you with the URL towards the whiteboard through the Zoom chat. So you just need to double click on it to open the whiteboard or copy the URL and put it in your web browser, and you will have access to this whiteboard. So in addition to the discussion, the brainstorming discussion, you can just add ideas on this whiteboard. You just need to Hello all. Hello everyone. Sorry, we made a slight mistake. We didn't come back all together in the meeting room to announce the coffee break. So I guess you have guessed that it's now time for coffee break until 4:30 roughly. So we meet you in 20 minutes time roughly. Sorry for the mistake. Bye-bye.
welcome back. Hope you enjoy a good coffee break. Even though we're not face-to-face, having a break is still fine during this long session on Zoom. So we'll go for the second session that will go on the same format than the first one. We're going to have two keynote speakers that will go for about 15 minutes and 5-minute questions, and then last speaker before the breakout session. the keynote, Alex Walden, who used to be at MBARI as well and now moved to Europe. And she will talk about the eDNA for marine biogeochemistry in general. So please, Alex, the floor is yours.
Okay, can you hear me?
Yes.
Yeah, and see the slides?
Perfect.
Yeah, okay, perfect. Well, thank you so much for inviting me. I really wish I was in Paris for this meeting. That would be really fun. But I moved to Europe and then COVID came, so I haven't gotten out and about very much. But I'm grateful for the opportunity to be here today and excited to talk to you about some of the constraints I see on eDNA for marine microbiogeochemistry, but I think we're in a really good position to move beyond those constraints. And my talk today will be largely conceptual. I think there are lots of folks here addressing some of the more technical sides, and of course we think hard about those as well. But I want to talk to you about some of the sort of fundamentals of biogeochemistry. What you're looking at here is a watercolor of this tiny organism Micromonas, which I was happy to see was abundant in one of Kevin's experiments that he showed. It's a tiny alga about the size of an E. coli in your gut, and around it you see seawater. But of course, when we talk about biogeochemistry, biogeochemistry is not about one organism. It's not about one photosynthetic critter. It's about the interactions between organisms. And that's where we really have our biggest challenges for application of eDNA. I would like my slides to move.
Simply try clicking anywhere. Okay, great.
Yeah, okay, thanks. So this is a rough schematic of some of the interactions we might see in the ocean. On the left-hand side, you see some of the photosynthetic organisms that are out there, and of course, very diverse, which we've learned a lot about from pretty early microbiology studies up right through many of the recent studies showing tremendous diversity there. And these organisms are consumed by heterotrophic protists. They are lysed by viruses, they might be attacked by parasites, and depending which of those interactions occur is going to shape the fate of their carbon. Does that photosynthetically fixed carbon ever really move up into the food chains humans like to think about that result in fish? Or does it ultimately end up being sequestered in the deep ocean? Another great fate for keeping the biosphere the way humans like it. Or being recycled in the surface ocean. So the current applications of eDNA are very powerful in terms of amplicon sequencing and metagenomics for providing a catalog of organisms present. Maybe, depending how deeply you sequence, you might have some rare players you can't always pick up. These kinds of studies enable us to look at diversity of microbes in the ocean or protists, and of course, gives information on relative abundance changes. So how does one template change in abundance relative to others? But again, understanding biogeochemical processes requires knowledge of activities, interactions, and fate. And so one thing I want to bring up, of course, is that oceans are seasonally and vertically dynamic. Even right out in the middle of the open ocean, we know well from coastal zones and our own experiences with winters that it's seasonally dynamic. But here you're looking from 0 to 200 meters out near Bermuda, and from the left to right on the x-axis, you're looking at the deep winter mixing period and the thermal stratification that occurs every year. And the big differences you see are how temperature moves through the water column. So these very warm months in midsummer. And you see this abundance of chlorophyll. So that's coming from the photosynthetic cells over in that deep winter mixing period. Now, winter happens to be a period that most people don't really want to go out and sample in the North Atlantic. So if we're surveying these sites and we're moving by when we can seasonally sample, we might miss a very important part of the community that can be quite different than the summertime community. So that's a really important consideration as we move forward in eDNA studies and application to biogeochemical process. So time series is essential for understanding biogeochemical processes. The other factor is that the nature of interactions and the players involved are often mysterious. So in this image, we have a diatom with its chloroplast shown in red there. And I think Chris Bowler might tell us this is kind of an unhappy diatom, but most of the rest of us don't know that. And what you can see are these bacteria interacting with that diatom. But we don't know what these bacteria are doing. Is it a friendly interaction? Are they colonizing a soon-to-be detrital diatom? Are they somehow a pathogen? And likewise, we can take our microscopes and look at protists and even see prey in their food vacuole. And we might know who those prey are, in this case, natural populations of Synechococcus and a Vibrio cholerae that's growing with GFP label in it, but we don't know who the protist is. So if we want to look at biogeochemical fate, these are things we really need to be able to connect, and that's hard to do in many of the common eDNA sampling contexts. So problem with pursuing uncultured organisms in nature, often sequences that we like to think of as providing evolutionary relationships don't reveal ecosystem roles or activities of cells. For example, whether an uncultured organism, we heard today about reference genomes and how important those can be. So in the case of uncultured protists without close reference genomes, is that organism a photoautotroph, a predatory mixotroph, so an organism that can do photosynthesis and eat, or a pure phagotroph? These can be things that are difficult to tease apart. And likewise, when we do size fractionated filtering with amplicon or metagenomic sequencing, it really captures a mixture of interactions. So we could be looking, when we filter onto a 3-micron filter, for example, we could be looking at bacteria that are prey and a predator, being the bacteria that are captured on that 3-micron filter. We could be looking at an ephemeral association between cells, as shown by Nicole King and some of her beautiful work on stimulation of sex and quantiflagellates by bacterial compounds. And we could be looking at a detrital lifestyle where there's a co-association on a filter because some bacterium is degrading that protistan organism represented by the big blue blob. So our aim is really to anchor co-occurrence analyses in demonstrated relationships between uncultured taxa. So what we'd like to do then is pair amplicon and metagenomic derived diversity information with tracking of microbial interactions in the wild. And to do that, we're going to be using fluorescence activated cell sorting, or FACS, and stable isotope probing, referred to as SIP. So for FACS, we'll be doing single cell sorting for cells stained with a stained food vacuole, so cells that were actively eating when we sorted them, or a food vacuole with chlorophyll, and then we know that they're a predatory mixotrophic organism, an organism that can do predation and photosynthesis. Now, we've seen in some of the earlier talks sort of some developments along the way as sequencing technologies changed. And I'd like to say that early on for my lab, what we were able to get out of these sorting approaches were really insights into evolution and diversity for protists lacking reference genomes, but we weren't really able to get a lot of ecological information because, as you all know, lots of proteins are of unknown function. And so you sequence away and you don't necessarily get the bottom line on ecological activities, but you can get a lot on evolution. For the stable isotope probing, what we've been doing is incubations with C13 and N15 labeled realistic prey. So in this case, we use the most abundant photosynthetic organism on the planet. That's the Cyanobacterium prochlorococcus. And we use these PQ eukaryotic cells like that one I showed you in the watercolor on the first slide. And then we're going to sequence any eukaryotic organism that seems to have incorporated that label, meaning it has consumed that prey item. So when we go out and look at the whole seawater with 18S rRNA in this case, what does it tell us about the relative importance of different heterotrophic protists? So This is just an example of a bunch of different samples collected at a coastal site, and these are very coarse delineations. But you know, you get out this list of how a bunch of different heterotrophic protists changed in relative abundance over time or sites or whatever it is that you are sampling for. Now, what does single cell sorting tell us about important predatory protists at the same sites? So here what you're going to see on the y-axis is green fluorescence representing an active food vacuole. And on the x-axis, you're looking at forward angle light scatter, which is a rough estimation of cell size, or indicator of cell size, I should say. So that group of blue dots there are cells with protistan cells with active food vacuoles. On the right-hand panel now, you see chlorophyll fluorescence on the y-axis and forward angle light scatter again on the x. And in this case, what we've done is excluded any of the cells that include photosynthetic pigments. So we just want to look at the pure predators. And what you can see is that 99% of these cells that we sorted were an uncultured choanoflagellate named bicosta. So those choanoflagellates were part of those prior whole filtered seawater samples that we looked at, and I'll show you that up a little more closely. First, I want to add that choanoflagellates are really fascinating because they're among the closest living unicellular relatives of animals, and they have a unicellular state that we're sorting, and they have a truly multicellular state, not just colonial. So they're often used to look at the evolution of multicellularity in animals, in addition to being predators in the ocean. So when we look at choanoflagellates in the amplicon data from filtered seawater, they're in yellow here. And you'll see that the yellow little fractions on the y-axis, you have percent of phagotrophic, heterotroph amplicons. You'll see that they're really very low in relative abundance compared to many of the other predatory taxa. Now, our FACS data was telling us, but they're really active out there. So, you know, and they're seemingly abundant. So why aren't we seeing more of that? So what we wanted to do is use another method to confirm this. And we used stable isotope probing with these labeled prey. And what we could see, and this is moving out towards more open ocean water, is that the taxa that incorporated these prey were the marine stromatophiles that are uncultured, some chrysophyceae, and then again, these choanoflagellates. So our activity-based assays are telling us something a little different than relative amplicon, and that's likely to do with differences in copy numbers, et cetera, that are known for the 18S ribosomal RNA gene. So activity levels are not necessarily well represented by relative amplicon abundance in this case. But we can use those same experiments to look at the inner world of uncultured choanoflagellates by doing that single cell sorting, then doing whole genome amplification, sequencing, and doing genome assembly. And in this case, again, we're looking at bicosta minor and uncultured choanoflagellate in 99% of the wells based on 18S. And what we captured with it was a giant virus, an uncultured giant virus. Now, many uncultured viruses have been seen in eDNA from the ocean. Early clone libraries, metagenomic data mining, metagenomic sequencing and assembly of aquatic giant viruses have all exposed them. Lots of other studies, including some of the early studies on giant viruses and moving into the most recent studies. But the true host of most of these viruses, even if you can assemble them, is not clear unless you have a very close reference, cultured reference virus. So this uncultivated choanoflagellate virus is giant. It has a 900 kb genome and 22% GC. You can see it down here close to mouse, which is of course kind of like us. And here's the only other virus known from marine samples from a predatory protist. And of course, it's in a very different evolutionary group than the one we've captured. Now, there are lots of other giant viruses known for aquatic organisms, lots of them from algae, which tend to be better represented in culture collections. And I want to emphasize this is a really fast moving field, so there might be some studies that aren't on this slide yet. One thing I did want to point out from the technical side is that for the whole genome amplification, it really matters what enzyme you're using. So what you're seeing in these plots is a comparison of the same sorted cells or multiple replicate plates of the original enzyme, the wild type 529 that was available. You're seeing it with the new WGAX mutant. And what you can see in this %GC plot, which is what you're seeing along the x-axis, that the old enzyme captures low GC templates better. The newer enzyme barely captures them at all below a certain threshold. So when we look at %GC content of some of the latest giant viruses, you'll see that they wouldn't even be captured by the new enzyme. So we do have to pay a little bit of attention. We learned this through some very unfortunate attempts to confirm our initial studies where we moved to the new enzyme not knowing about this and then realized why we weren't seeing it. But anyway, something to think about. Now this flagellate virus, again, large genome, these kind of stats in terms of the proteins and what they affiliate to are very common for giant viruses. Some of the neater things were that it has these orthologs shared with cellular organisms, photolyases, translation initiation factors, aminoacyl tRNA synthetases, but it also has 3 microbial rhodopsins. And at this point in time, only one other is known from a virus with a known host, and that's of the alga Phaeocystis. And although other putatively viral versions of this gene had been seen in metagenomes, so microbial rhodopsins, of course, can be used as proton pumps or a variety of other functions in microbes. And what you can see here is that this one is really divergent from the ones known in archaea and bacteria. And what we've been able to do with this approach is sort of attach hosts to some of the environmental sequences where before we only had the one known from Phaeocystis. So hosts for those in eDNA samples are unknown without reference data. And if you can't get the thing into culture, maybe because it's a predator and needs a specialized prey item from the field, you will never learn about it unless you take on some of these other environmental approaches. And this is just to point out that the hosts actually also have these genes, but that is not the one that's in the viruses. It's an ancient gene from viruses, transferred some other way sometime long ago. Now, this means that we can start to take something like this plot from Tara-Ocean's data, where you're looking at the frequency of a marker gene for these kinds of viruses and the frequency of that viral rhodopsin, and think about what it means in terms of the presence of this gene in other viruses. And you can see that it's not quite a one-to-one, but that this gene is likely in many marine viruses and possibly a core element of marine giant viruses. So these expose new aspects of biogeochemical exchanges and controls of predatory protists that we can now go and examine in eDNA studies. You see viruses in host, and a story I can't tell you about today is, in the interest of time, which I'm running out of, our intracellular non-prey bacteria that we're finding, which we think are symbionts. So this is really important for marine biogeochemistry because these interactions involve direction of carbon and energy flow. So when we go to model an ecosystem, we can't just know about diversity and dynamics. We have to know about direction of energy flow. And last, I just want to say, touching on a point that was brought up in one of the earlier talks, the importance of bringing statistics to marine microbial eDNA analysis. And in this case, we're moving into the sediments using a remotely operated vehicle. And we're comparing multiple replicates of deep sea sediment cores. And here's a deployment you can see where we're really doing the randomized sampling that you would typically do in terrestrial ecology studies, but it's not very common yet in marine studies for all the reasons we heard about: cost and difficulty and deployment and time. But what you're seeing here are multiple sediment cores where we're going to do randomized pulling to look at the microbial communities and replicate sampling within each of those cores. And for a subset of these, we've put in a preservative that should prevent the signal from changing as we move up to the surface of the water column. And I have more data on that, but I don't have time to show it to you. I do have it in some backsides. We could talk about it if anyone wants to. And with that, I want to thank everyone in the lab that makes these kinds of studies so fun, and emphasize that I think we're at a really powerful point to integrate across amplicon or barcoding or whatever you want to call it, to process studies that involve connecting with the true biochemistry that's going on at that point in time in the ocean. And I want to thank you for your attention.
Thanks, Alex, for the great talk. We running late, but we can take 1 or 2 questions. Anyone? If none, we'll just move to the next speaker. Okay, so we'll move to the next speaker, so we save a bit of time. With Sophie Arnaud. We'll present Sophie, you're here, yeah?
Yes, I'm here, thank you. So I'm starting to share my screen.
Yeah, it's coming up.
I hope you see it. Do you see it?
Yeah,
okay,
perfect.
Thank you. So,
yes, Sophie, you can go.
Thank you. So it's going to be a little bit more basic and technical because I would like to talk with you about the sampling for eDNA-based inventory for benthic biodiversity in the deep sea. So we had a lot of very interesting talks about how to sample DNA from the seawater and how to process it and what we can do out of it. In our particular case, we were interested in the abyssal plain. As most of you know, most than 2/3 of the surface of Earth is covered by ocean, and more than 2/3 of the ocean of the ocean seafloor is lying below 3,500 meters depth. Here you have a very small bibliometric study showing that despite the vast majority of the ocean seafloor is covered by abyssal plains, most of the studies thus far have been working on high biomass concentration and fascinating ecosystems such as hydrothermal vents or cold seeps or cold water corals. In fact, one of the obstacles to study efficiently the biodiversity in the abyssal plain is the very low and heterogeneous density of life in the abyssal plains. So there are large uncertainties in marine biodiversity, and the deep sea is still a black box. If we look at pioneer works by zoologists in the 70s and up to the 90s, there's been a lot of very interesting pioneer works showing how diverse the benthic diversity can be in the deep sea and in the abyssal plain, in regional, one of the most important, regional analysis that was published by Grassel and Maciolek. It was only on the eastern coast of US. They could find in a very limited amount of sediment compared to what is there, 1,600 species, many families, and 40 phyla. And in recent reevaluation of marine biodiversity, there are very different let's say, estimates or prevision of the number of species still remaining to be described, and this can span from 300,000 to 100 million. And as has been underlined by our colleagues Paul Snellgrove, we don't know anything about their contribution to global biodiversity and biochemical cycles, mostly because we don't know them. And any of those very different estimates, given the amount we know compared to what remains to be discovered, are mere educated guesses. So the limitations mostly, and I'm going to be rather quick on this. The limitation, of course, is in order to describe benthic diversity, we need to process huge volumes of sediment. We need hours of trimming in the laboratory, often for microscopic taxa. And on average, it takes 3 weeks of human resources for each new species to be described. This is for the biological material and time resources limits. Now there's also a limit, that is the limit due to the observer effect in morphological inventories. Biodiversity inventories are made in different parts of the world in different moments in time. They are suffering an observer effect that makes them very hard to merge together in order to get a global picture. So eDNA was very quickly seen as a way to be able to have a standardized assessment of benthic biodiversity and to be able to merge different regional assessments in order to have a global picture of the benthic diversity in the deep sea. So I'm going to go very quick on this. This work is coming from the Ifremer project Pourquoi pas les abysses that was launched by Ifremer in 2016 and then became eDNA-based with the support of France Genomic and the Genoscope in 2017. And in this work, we wanted to focus on the benthic diversity on the seafloor as compared to all the big circumnavigations that had been done before, like Tara-Ocean and Malaspina. And we also wanted to insert the metazoans in the picture compared to the mostly microbial inventories that had been made thus far. So in terms of sampling, we had many wonders at the beginning of this work, and we were looking for a standardized, easy to deploy gear to allow for replicates. I'm a little bit ashamed after the previous speaker, because when I see what she calls replicates, I think we are far from it. But for the moment being, we've been working with 3 cores per sites, per station. And also what we wanted to do is, given the publications that have been mentioned in the morning by Philippe, showing that the DNA decay in the seawater is much quicker, we wanted to compare what we could obtain with sediment compared to the seawater that is just underlying the deep ocean seafloor, with the hope that because the seawater is kind of easier to process, we can pump huge amount of water and then try to integrate a large part of the present community. And because of the DNA decay in the seawater, we would get mostly contemporaneous community and try to avoid the pitfall of archives. Then we wanted to compare what we could obtain with sediment and with water. So this is mostly what I'm going to show you now. The water, we sliced also the cores into 6 different depths, because we know that the communities are strongly vertically segregating. And in any case, would the water be of help? It may help only to replace the first centimeter. This said, the first centimeter usually encompasses 70% at least of the biodiversity, at least in terms of metazoans. So what we did for processing and preserving the sediment is we compared sediment that would be sieved with sediment that would be not sieved. Of course, we did the slicing with sterilized spatules. We kept the slices in homogeneized sterile Ziploc bags, and we kept them at -80. The same was done for the sieved fractions. They were also kept in Ziploc bags and kept at -80. And so when we compared what we could obtain out of small volumes of water that are 2.5 liters, this is the white, the clear blue, and that could be obtained for several thousand liters filter. This is the dark blue. And what we would obtain with sediment, either not sieved, which is red, or sieved, that is kind of orange, what we could see is there was no way we could replace water filtering and DNA extraction in order to characterize the deep sea benthic communities, even of the first layer. And this is true for all compartments. Here I only show you the metazoan preferred markers that are 18SV1, 2, and CO1, but this was true as well for 18SV4 and V9, and for the 16S markers we used for microbial communities. So what we see is we have a different alpha diversity depending on the fact we deal with water or sediment, but more importantly that we don't reference at all the same phyla depending on whether we analyze water or sediment. And it's very clear that we had a very strong community differentiation between water and sediment samples. And this was mostly due to the fact that water, even again, very close to the bottom to the seafloor, was mostly allowing to reveal pelagic taxa, whereas all the benthic taxa remained mostly discovered through the sediment processing. So we stick to the use of triplicate cores sliced in order to characterize benthic community for sediment. And now we entered the moment that we actually apply the different we apply the molecular and bioinformatics to all the samples we've been gathering through this process, which is about 2,000 samples from the deep sea that have been collected thus far, in order to make a small synthesis of what we could obtain on only about 10 sites that are mostly in the Pacific over here and there. We obtained about 12,000 metazoans operational taxonomic units, so just with 30 cores. And we have several syntheses in course with our partner teams, and particularly with Tara-Ifremer. And we have one project that we hope will be funded in order to finish the analysis of all those data that were produced thanks to the partnership with Genoscope. And we are deeply involved in the bioinformatics and the community ecology analysis at the moment. So with this, I thank you for your attention, and I wanted to thank the dream team of bioinformatics optimization, and just to show you that bioinformatics is not only a male job. Here was the team. And I also wanted to thank the Genoscope for the partnership, and the CBMER with the bioinformatics cell of Ifremer for their support. So thank you for your attention.
Thanks a lot, Sophie. So it's time for the general discussion on the. If any of you have questions for Sophie or any of the previous speakers, you can ask them now. I suggest to keep that session a bit shorter than the 10 minutes already previously planned. Let's say maybe 5 to 10. Let's keep it 5, so we can break out then in the 2 different sessions. So Detlev? You're muted, Detlev.
Yes. Okay, Sophie, many thanks for an exciting presentation. I wanted to quickly follow up on the OTUs and this massive amount of species that you found. How did you deal with the ones with the unknown one? How do you proceed there further?
With which one? I did not hear you.
The unknown one, the one whenever you don't know.
This is excluding the one that were not assigned, at least to the phyla level.
Yeah, is there other new phyla? This is, I think, what I'm asking. Something that you really can't.
Well, I guess that's the growl. A lot of us are after, but it requires a lot of digging in the data and a lot of processing of sequencing. So at the moment, we are looking at the broad picture with what we know, and we are sure we understand. And certainly, the next step is to dig into them and to try to sort out whether the unknown sequences are actually complete paralogs and non-target sequences, or whether they are actually phyla, maybe new phyla or new lineages that are absolutely absent from the databases, which is absolutely possible. And at the moment, my main wonder is one of the things we can do with CO1 is try to see if we have an open data frame, an open reading frame. The issue being that for metazoans, this tends to change from one phyla to the other. So I'm not quite sure how to deal with it if we have unknown phyla. So if anybody has a bright idea, I buy it.
Thank you.
Thank you.
There was a question for Alex in the chat. You replied, but maybe you want to elaborate a bit about the interest of combining flow cytometry with eDNA to better understand interactions.
Yeah, I mean, I think it's a really powerful way to start to, Tara obviously did a lot of studies and you yourself on trying to really infer what interactions are going on, what relationships are going on between organisms. What we've found that's especially interesting with the cytometry is it often picks up on interactions we aren't seeing in the network. So that study isn't submitted yet, but the networks seem really good at looking at some of the more abundant characters, but not necessarily these very specific symbioses or viral interactions that could still probably have some quite important biogeochemical consequences. And someone, yeah, right. So that also answers, there was another question in the chat that I answered specifically to the person who asked it. Yes, we have done network analyses on the broader like size fractionated and whole seawater, again, using the algorithms and methods provided by Tara. And we do see some differences in what we're picking up.
Thanks, Alex. Anyone else? You can just raise a hand or step in. No? Okay. So it's good. We
Not
saving
enough
a bit of
coffee.
time.
We need
Yeah,
more coffee.
not enough coffee. Yeah. So I guess now we'll be soon split it in two different breakout rooms to continue the discussion. We were initially planning to have 30 minutes, but we're running a bit late. So maybe we can just do it in 20 and come back for the last talk of the day, starting a new session.
So what time do you want us back?
We should be back by 6.
Okay, so it seems almost everybody is back, minus evening effects, I assume. So yeah, so welcome then to the session number 3, the analysis methodologies. And I would like to go straight into the first talk, the first keynote by Jérôme, and on the interpretation of eDNA in the context of community ecology. Can you share a screen?
Hello, I will try to, yeah. There we go.
Great.
Thanks for having me. I'm not a marine ecologist. I will be talking, and I've been tasked to talk about community ecology. So I will give a brief tour of community ecology and the nexus of data. We've been talking a lot about data and about models. So we've all heard about different methods with environmental DNA, and I'm not a specialist, so I won't go in a lot of detail about this research program, which is so exciting and has us attend to all these very exciting talks today and the forthcoming 2 days. Basically, looking at extracellular DNA and its fate in nature and how we can detect it. But then the question arises immediately when you're an ecologist, what can you do with this type of data? And that has been, as Pierre Taballet was showing a bit earlier, there's been a lot of excitement on the side of ecologists discovering this whole new universe of data that was opening up, which may be summarized in this citation by ecologists, colleagues. If one dared to take 1 hectare rainforest, perform the molecular equivalent of placing it in a blender and sequence the resulting nucleic acids, what could be learned? Which is basically what we are all doing now. So the thought experiment has provoked a lot of reviews, a lot of theoretical insights, but the wake towards actual data that can be compared to model has been slow, predictably. And this is where we are now. So it's really an interesting time to be revisiting these questions. Community ecology is the study of how assemblages of organisms coexist in a given environment. Now, this spaghetti plot shows different concepts or words that are used in the British corpus of books over the 20th century. And you see, interestingly, that ecology was mostly a discipline of an organismic discipline in the beginning and the first half of the 20th century, with animal ecology and plant ecology being basically the only types of ecology that were being taught and learned at school by prominent scientists like Charles Elton and Tansley and so forth. And it's only in the early 60s that especially marine ecology went to a bump and back down. And the reason why the ecosystem ecology type is less fashionable today is because the scales of organization from population to community to global ecology have taken the fore in our thinking. And this is what we're seeing today. All of these aspects are very much in the background of a lot of the talks we're listening today, and probably in the next 2 days as well. So the start of community ecology, as we think about it, is it starts with Evelyn Hutchinson, who organizes this concept in a paradoxical form. Well, why can so many species coexist in spite of them competing for the same resource, basically? And so this paradox is, of course, not a paradox because many processes are taking place. And so community ecology is expected to predict a large number of patterns that we all know: species area relationships, relative abundance distributions, latitudinal diversity gradients, the diversity productivity, and the composition environment relationships, and the distance decay of similarity. All these patterns, however, have to be explained somehow by some sort of theoretical framework that would encapsulate these different observables. And this is what I wanted to reflect on today. And of course, not to forget that community ecology is tightly linked with conservation biology and other forms of biology, population biology, and also, quite importantly, environmental management and political science. This is, of course, a lot of what we're trying to do today has to have somehow a societal consequence, and this is how theory could also help in forging consistent concepts. So the main critique of community ecology has been that at the global scale, it was easy to think of latitudinal diversity gradients. At local scale, in population, it was also clear to understand and make clear predictions. But at this mid-scale of the community ecology, where several species, an unknown number of species, interact in an unknown type of relationships, it becomes a mess. And this is how John Lawton summarized community ecology. Community ecology is a mess with so much contingency that useful generalizations are hard to find. And I want to perhaps prove John Lawton wrong, although it's been 20 odd years ago, and there is a lot that has been shown, not only in terms of the data, but also in terms of theory. One of the way forwards is summarized in Mark Villen's review paper on theory in community ecology. Basically, what community ecology does is takes a bunch of processes which are really inherited from population biology and population genetics, selection, drift, speciation, dispersal, and tries to make all these predictions that I was talking about a few slides before. Now, a more canonical way of looking at this fairly obscure black box theory is to look into the box. And if you, I have this, sorry, going back, yeah. If you look at the basic processes, speciation, it mostly occurs at the regional scale. Dispersal is what occurs especially what occurs when the species migrates into the sample that you are actually sampling. And within the sample is a fraction of the ecological community, which is a selection or an environmentally filtered fraction of what is out there. So selection plays a role here. And then, of course, what you sample, as Pierre was saying earlier, can be slightly different from one sample to the next, even almost at the same place. So that means there is some drift and some demographic stochasticity, environmental stochasticity. All of these processes are important to take in mind. So this is a bit of what's inside the community theory black box. And so one of the major ways forward is to take away selection and to construct a theory of biodiversity, a theory of community ecology that does not take into account species differences. So when you try to do this, Steve Hubble was working mostly in terrestrial systems, has called this the neutral theory of biodiversity. You cannot make all types of predictions because the relationship between composition and environment and between diversity and productivity cannot be predicted by this type of theory, but you can make an awful lot of predictions, and I'll show a few. One of them is, so one of the questions we've been asking is how well can you predict the parameters of the neutral theory of biodiversity using environmental DNA data, given that these data are not error-free. They have a number of types of biases, which may be realistic due to PCR noise or due to including more motifs, more operational taxonomic units that may be imagined. And one of the patterns is that if you look at the panel A here, you see that most of the predictions you make in terms of relative abundance distribution are actually quite similar, even if you take into account quite a lot of error. But the predictions of the parameters in the neutral theory are biased quite early on. In contrast, taking into account the PCR noise, the error noise due to PCR in metabarcoding, creates quite a different shape of the rank abundance curve, but actually does not modify too much the estimator of one of the parameters of the neutral theory. So that means that for there is an acceptable level of error that we can adopt, below which the theory is quite robust to changes and robust to error, which is a desirable feature for theory if you start to compare theory and data. Another example I wanted to share with you is taking us quite a bit away from tropical from marine systems and into tropical forests. Been working with colleagues in French Guiana in the northeast of the Amazon in this CNRS research station called NURAG, and we have sampled 12 hectares of forest every 10 meters. It was a project led by Pierre actually, and he's here actually making sure the samples are being collected right and being super careful with this. The end results of this pretty ambitious project was maps of diversity over this 12 hectare. Every 10 meters we have a sample, and we've been able to extract DNA from all sorts of the tree of life, from archaea to bacteria and to eukaryotes. And it's quite interesting to ask the question of whether we can predict some basic features of biodiversity. One of the results from Lucy Zinger's paper a couple of years ago, which is summarizing these results, is that the neutral theory predicts actually quite well the distance decay similarity, the rate at which species decay in similarity when you walk further away from a focal point. And this is irrespective of the taxon you're looking at. So that could mean two things. One of them is that the distance decay similarity is actually not really a robust metric to detect the departure from neutrality or the departure from any model for that matter. It's not an information-rich metric, or it may mean that the behavior of biodiversity is actually quasi-neutral. Moving away from the neutral model of biodiversity, the niche model of biodiversity makes almost opposite predictions and is actually quite interesting to predict the composition-environment relationships and the latitudinal diversity gradient. So just to show you a couple of examples here, one of them is predicting the relationship between the same type of data, the same data set I was showing just a couple of slides ago, with the environmental features of the soil. So we're really talking about soil metabarcoding here. And you see that this is not the easiest diagram to see here, but every piece of the cake here is a partitioning of the variance in biodiversity for every group. And we are asking whether the abiotic components, plants, or the spatial components, or combinations thereof are more important. And actually, we don't have a very strong result to show, except for the fact that some groups are much more explained by the environment than others, meaning that it's quite likely that actually neutrality would be at the scale anyway, more likely than in some groups than in others, which kind of makes sense. But this is interesting to have a quantification of these features. Another feature is the latitudinal diversity gradient, and Noah referred to the Tara wonderful recent work published on the latitudinal diversity gradients, and you see that predictions of these gradients can be made by theory, basically that higher productivity ecosystems are also the ecosystems that are more, they have the more diversity. However, let's not forget that the words of John Lawton, that these global biodiversity patterns are the easiest and the ones that we can predict with the least problems, because there is less contingency going on. It's not to say that this paper is not just super nice. Okay, another interesting feature, I think, in light of the discussion today, is how do we infer in interactions across groups of organisms based on co-occurrences. And this I think is a very important feature for this workshop. We tend to confound the effect of the biological interactions with co-occurrences. And really at the core of this is the fact that when you find two species or two motifs in the same sample many times over, then you suspect that there is something going on. But from this suspicion to the proof, there is always a long, or often at least, a long journey. And we've been using a different system in the Alps with a group led by Wilfried Thuillier in Grenoble to try to disentangle the groups and their interactions, statistical interactions between functional groups. And you see that the result is quite unexpected. The major contributor and the major relationship between the environment and biodiversity is actually through the springtails, not through plants. And this is not something we would have expected. This technique is quite interesting, and I refer to the paper in Ecology Lectures a couple of years ago. So to finish, I would like to speak a little bit about the limits to the use of eDNA. One of them is the relationship between motifs and species. And I won't spend a minute on this because a lot of people have thought about this. And the bottom line is that we need excellent reference libraries. And this is something that a lot of you are advancing, and this is still not done, but it's quite important. Second topic is individuals in ecology and of course evolution. And the third one is really briefly ecological resistance and resilience. So on the second point, if you look at any theory in ecology and also in evolution, what is being selected, what is being dispersed, what is drifts, and what speciates is really around the concept of individuals. In eDNA studies, we don't really have individuals. We have these bags of genes which are dispersed, open, split open and dispersed in the environment. And this is what we see. But from those bags of genes back into the concept of individuals, there is a long way. And that there is actually a large literature in ecology looking at the ecology of individuals, trying to do a real sociology of species other than humans. And I think there is an interesting way forward to think about how we can connect eDNA and ecology. The other points are the issue. The last point is the issue of ecological resistance and resilience. This is really a topic of foremost interest and importance in society. And if you look at medical studies, you see that there are quite a lot of studies where following individuals through time with massive sequencing projects every day almost. And this time series shows how fast you can recover from, living abroad or from diarrhea, which is not the most exciting thing to be undergoing. But this is really individual resilience. On the very long term, Esker Villeslevs or others have seen the resilience of biodiversity through time, through the last glacial maximum. I would like to argue that there is a kind of a shortage of data on the intermediate scale of where we can monitor on a monthly basis for years at a time the resilience of ecological communities using eDNA data. And I'm sure many of you this is a dear project to many of you, and this is something that we need to put to the fore, the Genomic Observatory or the likes, and try to have this long-term series and explore early warning diagrams of resiliencies or lack thereof. So in conclusion, there's been much hope for that eDNA would transform community ecology. Unfortunately, we're not quite there yet because of conceptual differences in the way we are thinking about biodiversity. Tests of community ecology theory using eDNA are likely to be limited by key assumptions, and that we need to talk about. And finally, I would like to reemphasize the central challenge to have a good temporal time series and based on good data. And I'm sure that this is something many of you share. Thank you very much.
Thanks, Jerome. Any questions? Then let me start with one. So very nice talk, but it was all on a biotic interactions in a way. But at the end of the day, ecosystems include lots of abiotic factors. And the problem I see is that so many, and it's hard to figure out what are the important ones and how big are they compared. So in a macroscopic world, if one species eats another, has a big impact. If you go down in size, then the chemical factors become probably more and more dominant. Temperature, a good example. So a given network only has a temperature range, it can survive or be resilient, etc. So how would you factor this in, the abiotic part, by lots of external, let's call it metadata monitoring longitudinally, or how would you get this in?
Yeah, thanks for the question. It's an important point. Of course, quantifying the environment of these ecological communities is, of course, a crucial aspect. I was under the understanding that it was easier still to measure temperature, to measure eDNA, and so these environmental features are being monitored and are being measured on the technology exists already, and I don't think this is conceptually difficult to the extent that the eDNA and ecological communities are difficult to quantify. So this is why I focused on the biological aspect, because I think this is where the nut is the hardest to crack. But I agree with you. It makes no sense if we don't have the abiotic environment and co-quantify the abiotic environment.
Thanks. I think Bruno was next.
Yes, thank you. I was particularly interested in one of your last comments on individuals in ecology and the fact that based on eDNA, it's really difficult to get down to the individual level. But do you have already any ideas of how this could be done or how to progress in that direction?
I suppose that a dramatic departure from the accepted dogma in community ecology that to take away individuals and to accept that we are looking at something else, or the other metrics, would be interesting. That would be an interesting direction. So that would be on the side of community ecologists to move forward in this direction. The other way around is, of course, you cannot exclude that some progress will be made to quantify this link between individuals and eDNA. And I'm sure many of you have ideas, and it may be also system dependent. It may be easier in some groups than in others.
Thanks a lot. I think Pierre was next.
Thank you, Pierre. Very interesting talk. Thank you, Jerome. So one question, or one thing that keeps popping up with ecology in general is, of course, scale factors and patchiness. And in marine microbiology and even in sedimentary systems, we cross many of these scales. It's very hard to characterize what kind of patchiness we look at, etc. What's your perspective on this? How do we cope with this? Is there a sampling strategy we do, pilot studies to normalize it? How do we move forward in that?
Well, conceptually, the problem of scale is really embedded into the theory. This has been a major challenge in community ecology for a long time. So those theory do take into account this scale, both in scales of integration and spatial scales. Of course, I cannot answer the second part of your question because this is way too system dependent. Exactly how do you sample well your environment spatially? I believe this is a trial and error approach. There is no general rule. And I on that matter, I really agree with Pierre when he was mentioning that pilot studies are really needed in every step of the way.
Thank you,
Thanks.
Jerome. Yeah.
Okay. I think Chris was next.
Okay, thank you, Pierre. Thanks, Jerome, for that very thoughtful talk. I would just want to pick up on the comments you made about the importance of individuals in ecology. And yeah, you know, that's very much a sliding bar. I see there's also some discussion in the chat about this, that, you know, if you're thinking of individuals in terms of elephants or human beings, it's obvious, it's clear what individuals are. When you're talking about a colony of ants, then maybe the individual is the colony, right? Not the individual ant. So when you get down to microbes, what is the concept of individual? Where are we? Maybe different for depending on what you are. If you're a protist with some sort of behavior, the individual protist may have some relevance. So yeah, you've thought about this more than we have probably. So I'd just like to have your further thoughts about why this individual concept of individuality can be of relevance in marine ecology.
Thanks, Chris. Well, there is obviously a historical feature of ecology being an animal and plant ecology at the beginning, so macro-organisms. And if that matter, individuality makes perfect sense. I know some have discussed the fact that microbiology doesn't fit in many ways, and even in evolutionary biology, you know, certainly Carl Woese discussed this many times over. So this is one point. Now, when I'm focusing on the individual, let me be clear that I never say that there is an average individual. Individuality and differences in behavior, in physiology is actually really part of all this community ecology. And this is something that ought to be quantified, which is what population ecologists also do. So these aspects need to be somehow reconciled. But I agree with you that when you go down to the microbial world, things don't go squarely according to theory. And this is something that has been looked into, but not too much for lack of shared I think shared theory between microbial ecology and macrobial ecology. There have been some attempts, but I think that these have been mostly sideways. They haven't been quantitative theories. There have been more conceptual ones. So there's a lot to do.
Okay, thanks. We are running a little out of time, but maybe Raffaele, you have a last question then.
Yes, thanks, Pierre. Jerome, thank you very much for this nice talk. Of course, I agree with you that long-term monitoring systems and global scales studies are useful to study community ecologies. But I was wondering about your opinion about the use of complementary approaches, like, for example, mesocosm experiments or common garden experiments or statistical modeling for studying community ecology using eDNA, environmental DNA. How far we are, for example, for statistical modeling to integrate eDNA data and to study community ecology, according to you?
Well, of course, there's been quite a lot of efforts at European scale anyway to construct some experimental setups to test ecological theories. And this is really quite exciting. There was one in Paris, one in the southwest of France, nearby where I am. And those systems are trying to control for environmental features and test different types of hypotheses, all related to the ones I was showing. This is, of course, complementary and by no means conflicting with observing the systems. In my view, you cannot test a hypothesis if you don't have the hypothesis in the first place, and if it's not well formulated. And so this is quite important to go first to the field, whatever it is, to go and formulate the hypothesis in the first place. But the tools and instrumentations like echotrons and so forth are in place to do exactly that. Thanks, there was.
Okay, thanks again. I think Pierre's question partially overlapped with Chris's question, so you can consider and conclude. It was a great talk again, and seeing everybody tomorrow, 2 o'clock. So, with continued excitement, very cool stuff. Okay, thank you, everybody. Bye.